So welcome to the first high-level unit for this topic in biochemistry, proteins and enzymes, which will extend on the SL topic of the same name. So by the end of the lesson, you should know that inhibitors, or what inhibitors are, linking amino acids and proteins to the buffer work we've done earlier in the course, look at the protein assays that are used for proteins and enzymes, and should be able to determine Vmax and the Michaelis constant, Km, using graphical means, compare competitive and non-competitive inhibition of enzymes, explain the concept of product inhibition, calculate pH values for buffer solutions, which again we've already looked at, and determine the concentration of a protein in solution from a calibration curve using something called Beer-Lambert's law. So, first thing, the chemical composition, structural features and biological functions of amino acids and proteins and enzymes have already been discussed in this topic earlier in the SL component. And the study of the activity and distribution of these compounds in living organisms is a key area of modern chemistry. The role of inhibitors and regulation of enzymatic processes, quantitative interpretation of biochemical data, and spectroscopic techniques used in protein analysis will be what we're looking at in this lesson. So the first thing we're going to look at is amino acids acting as buffers. So we've already looked at acid-base equilibria, and basically, two amino acids can act as acids or bases depending on the particular pH of the solution. The carboxyl and amino groups of these compounds can be ionized in various extents, producing ionic species. Remember the zwitterions. In summary, in strong acidic solutions, amino acids and proteins are protonated and can exist as cations. And in strongly alkaline solutions, deprotonation occurs and anions are formed, as you can see from the diagram on the slide. At a certain pH, known as the isoelectric point, which we've already looked at, this one here, basically the pH will equal the isoelectric point. And this is where you have the zwitterion, where the positive charges and negative charges are equal to each other. And it's going to be specific for each amino acid or protein. And the positive and negative charges of the ionized groups cancel, produce the zwitterions, which we already looked at, with zero net charge. So looking at this in more detail, each of the two equilibria, okay, which we look at here, okay, there's going to be two equilibria happening, okay, 1 and 2, okay, the cation zwitterion and the zwitterion anion in the above scheme involve a pair of species differing by a single proton. Okay, H+. Such pairs are of course known as conjugate acid-base pairs, which we've already looked at, where the more protonated species is the conjugate acid and the less protonated species is known as the conjugated base. Okay, so look at this, it'll be the conjugated acid here, and that'll be the conjugated base here. An equilibrium between the components of the conjugate acid-base pair is characterized by a dissociation constant. Okay, so here we have basically, for amino acids, two dissociation constants. So the cationic forms of two amino acids with non-ionizable side chains have two acidic centers. Okay, so you've got the COOH, the carboxyl group, and of course the NH3+, kind of amino group there. Okay, therefore there will be two dissociation constants, pKa1 and pKa2, which we see on the slide. The carboxyl group has a relatively high acidity and dissociates more easily than the protonated amino group. So, the pKa1 value characterizes the equilibrium between the anion and the zwitterion. Okay, between here, so between the cation, this one here, and the zwitterion here. Yes. Okay, the pKa2 value refers to the equilibrium between the zwitterion and the anion of the amino acid. Okay, so zwitterion, okay, here, and the anion of the amino acid. Note that the same zwitterion in the conjugate base is the first acid-base equilibrium, but the conjugate acid in the Note that the same zwitterion is the conjugate base in the first acid-base equilibrium, but the conjugate acid in the second equilibrium. That's an important point to note. In any aqueous solution, only 2 of the 3 possible forms of an amino acid can be present at the same time. One of these forms is always the zwitterion, while the other form is either the cation or the anion. Both the cation and the anion of the same amino acid cannot exist in the same solution because they would immediately react with another to form as zwitterions, which we see from these equations here. Okay. Therefore, acidic solutions where the pH is going to be less than the isoelectric point contain mixtures of cations and zwitterions, while alkaline solutions where the pH is greater than the isoelectric point contain zwitterions and anions. And the exact ratio between these two forms depends on the solution pH and the pKa of the associated conjugate acid that is present in solution. And it's where we get to the Henderson-Hasselbalch equation, which we've already looked at previously in the acid and base section of the course. Since pH equals negative log base the proton concentration, the pKa expression can be transformed into the following Henderson-Hasselbalch equation. At pHs less than the isoelectric point, the conjugate acid is the cationic form of the amino acid, the conjugate base is a zwitterion, and the pKa equals pKa1. At pHs greater than the isoelectric point, the conjugate acid is the zwitterion, the conjugate base is the anion, and the pKa equals pKa2. Now, the advantage of the Henderson-Hasselbalch equation is that it allows calculation of the pH of an amino acid solution with a known acid-base composition or the concentration of a conjugate acid and base in a solution with a known pH. Now, the advantage, of course, is that you can start making acid-base buffers. So an acid-base buffer solution or buffer solution contains a weak conjugate acid-base pair, just like the amino acid examples we've got, and they can neutralize small amounts of strong acids and bases without significantly changing its pH. In an amino acid buffer, the strong acid is neutralized with the conjugate base of the buffer, while the strong base reacts with the conjugate acid. So let's look at some examples of how we can apply the Henderson-Hasselbalch equation. So first one is calculate the pH of an aqueous solution that contains 0.8 molar zwitterionic and 0.2 molar anionic forms of serine. Okay, and you've been given both the pKa1 and the pKa2 of serine and also its isoelectric point as a little bit of a red herring. Right, so this is the answer. So the solution contains anions already, okay? So we know that the pH is going to be greater than the isoelectric point. It's going to be alkaline because you've got the proton being removed. So that means we're going to use the second pKa, the zwitterion in equilibrium with the anion and a proton. Therefore, the zwitterion is the acid and the anion is the conjugate base. We go to our data booklet, plug in the numbers from the equation, or into the equation, and you get the associated pH, which is alkaline, of 8.5. Right, next question. Calculate the pH changes of the addition of 1 gram of solid sodium hydroxide to 1 decimeter cubed of pure water first up, and then 1 decimeter cubed of a buffer solution containing 0.4 molar of a zwitterionic and 0.16 molar of a cationic form of glycine. And the assumptions are that the density of water is 1 kilogram per decimeter cubed, and solution volumes do not change with the addition of the sodium hydroxide. This is where, of course, now the amino acid solution is acting as an acid-base buffer solution. So again, we've been given the pKa's, associated pKa's of the glycine and also its isoelectric point. Okay, so the first thing we have to do is work out the concentration, the pH of a sodium hydroxide solution of 1 gram being added. Okay, so we work out the number of moles of sodium hydroxide, which will be equal to 1 mole of hydroxide being formed, because that's what happens when it dissociates. Therefore, the concentration, the number of moles of hydroxide is 0.025 mole. The pOH is going to be negative log base of the concentration of hydroxide, which we've just discovered, which will be 1.59. So actually that should be 2 decimeters cubed there. Oops. Okay, 1.59. pKw equals pOH plus pH, so we can determine that the pH will be 12.4. So pH water is 7 and change to pH of 12.4, so a pH change of 5.4. Now let's discover the effect of adding an amino acid buffer system to the change in pH. So the solution contains cations, therefore it's going to have an acid pH due to the protons being added. So we use the pKa of 2.3, zwitterion plus a proton going to a cation. Therefore the zwitterion is the base and anion is now the conjugate acid. So again, the sodium hydroxide associates into sodium ions and hydroxide ions, and the cation then reacts with the associated hydroxide ion formed to form the zwitterion plus water. Then we make a nice little table where concentration equals number of moles divided by the volume, where the volumes are nicely all 1 decimeter cubed, just for you. Okay, so we do the Rice method. Okay, so the ratio, okay, is 1 to 1 to 1. And then the initial concentration is going to be 0.16 of the cation, 0.025 of the hydroxide, and 0.4 of the zwitterion from the question and from our previous answer. Calculate the concentration, okay, and it's going to lose 0.025 moles of the cation and hydroxide, and the zwitterion is going to increase by 0.025 moles. Therefore, the end is going to be 0.135 of the cation, no hydroxide left, and the zwitterion will be now of the concentration of 0.425. Then from our data booklet, we plug in the numbers from the equation, and we get a pH of 2.7 at the start, and then a pH of 2.8 at the end. Therefore, only a pH change of 0.1. Okay, and this just essentially shows the effectiveness of a buffer to resist pH changes. And the final problem, identify the conjugate acid and conjugate base of a 0.5 molar solution of glycine at pH 5 and calculate the concentrations of both glycine species, okay, the protonated and the deprotonated. Okay, so pH solution is below the isoelectric point, so it contains cations, okay, yeah, pH there, isoelectric point. Therefore, the pKa of 2.3 must be used, okay, because we're now in acidic solution. So the zwitterion is reacting with a proton to become the cation. Therefore, the zwitterion is the base, and the cation is the conjugate acid. So again, from the data booklet, we plug in the numbers. Okay, so it means that the zwitterion concentration compared to the cation concentration ratio is 501, quite large. So if the cation becomes X, then that means the zwitterion must be 501 times the concentration of the cation. So that means the total concentration is going to be X, is going to be 0.0001 molar. Now, we already know this is not an effective buffer because the pH is more than pH 1 value away than the pKa. So it's not going to be actually quite effective as a buffer. So we're now just focusing more on the enzymes, okay? So what are enzymes? So basically, this is just a summary of the previous slides from the SL component. Okay, so the basic concepts of enzymatic reactions were discussed previously, so in the SL component, so look over that. And one of the original theories, the lock and key model, described the process of chemical recognition between the enzyme, which acts as the lock, and the substrate, which acts as the key, as an exact fit of their complementary structures. Although this model can account for the specificity of enzyme catalysis, it was unable to explain certain experimental data, in particular the enhanced ability of transition states in enzyme substrate complexes. However, the development of X-ray crystallography and computer modeling systems allowed the three-dimensional shapes of active sites in enzymes to be determined, which in many cases did not match the shapes of their substrates. So that's kind of led into the idea of the induced fit theory. Okay, that's why it was superseded. Experimental data said, well, your theory about the lock and key doesn't quite match, so we need to kind of change the theory and hence we had the induced fit theory, first proposed in 1958 by Daniel Koshland. So, essentially, the way it works is that you've got basically two forms of inhibition. And the induced fit theory also explains the mechanisms for enzyme inhibition and activation that regulate the metabolic processes of living organisms. Apart from their main active sites, many enzymes have additional allosteric sites that can be temporarily bind to specific molecules via weak non-covalent interactions. Okay, so basically you see it on the side that substances can bind to an enzyme outside the active site to inhibit its effectiveness. Okay, so here would be an example right there. When an allosteric site is occupied, the shape of the enzyme molecule changes, which alters the configuration of the main active site. Just showing the power of 3 dimensions in the enzymes. This in turn affects the stability of the enzyme substrate complex and the ability of the enzyme to act as a catalyst. In most cases, allosteric interactions reduce the enzyme's activity, which is known as allosteric or non-competitive inhibition. The term non-competitive refers to the fact that the substrate and the inhibitor have different chemical structures, bind to different sites of the enzyme, and therefore do not compete with one another on the main active site. Now, the other form of inhibition is competitive inhibition. And this takes place when the substrate and the inhibitor have similar chemical structures. In this case, the inhibitor may occupy the main active site and prevent the substrate from binding to the enzyme. The most common type of competitive inhibition is product inhibition, which is quite useful. This is where the active site of the enzyme is blocked by the product of the actual enzymatic reaction. Product inhibition may also occur via the non-competitive mechanism, where the reaction product binds to the allosteric site and reduces the enzyme activity. Okay. So that's essentially an idea of a negative feedback. So both competitive and non-competitive product inhibition provide negative feedback to metabolic processes, which is the bioequivalent of Le Chatelier's principle, which we've looked at previously when we looked at equilibrium. When the substrate concentration is high, the rate of the forward reaction increases. Okay, so the concentration of the substrate is high, the rate of the forward reaction increases. Okay, and the excess substrate is metabolized. In contrast, with a high concentration of product, okay, the concentration of the product increases. This inhibits the enzyme and prevents further increase in the product concentration until it turns to normal. So it kind of favors the other direction, like Le Chatelier. So the rates to many enzymatic reactions were briefly mentioned in the SL unit around the enzymes, and they can be described by the Michaelis-Menten equation. Okay, so the Michaelis-Menten equation is V equals V max, concentration of the substrate, divided by Km plus the concentration of the substrate, where V and V max are the actual and maximum reaction rates respectively. And the concentration of S is a substrate concentration, and Km is known as the Michaelis constant. And the Michaelis constant is equal to the substrate concentration when V, this guy here, is equal to half of V max. The values of V max and Km depend on the enzyme concentration, okay, which is the, there we go, that way there. So the Michaelis-Menten equation can be applied only when the concentration of the enzyme is a constant. Okay, so when the substrate concentration is low, so Km is going to be way higher than the concentration of the substrate, Km plus S will equal Km. Okay, therefore V will equal Vmax over Km times the concentration of the substrate, which corresponds to a first order reaction. At low concentration of the substrate, almost all active sites of the enzyme are available for the substrate molecules, so the reaction rate is proportional to the substrate concentration. However, as the concentration of the substrate increases, more and more enzyme molecules combine to the substrate and form enzyme-substrate complexes, which reduce the number of available active sites. Okay. When all the active sites are occupied by the substrate, the enzyme works to its maximum capacity and is said to be saturated. Any further increase in the concentration of the substrate will not affect the reaction rate because additional substrate molecules will have to wait until active sites become ready again. Okay, this is shown in the Michaelis-Menten plot as a basically a first order reaction. So you've got your linear increase here, so that's going to be your first order. Okay, and then as you get saturation, okay, saturation of the active sites, you get a zero order reaction. Okay, so since Vmax is limited to the number of available active sites, it must be proportional to the enzyme concentration. So Vmax will be equal to the catalyst equilibrium times the concentration of E, where Kcat, known as the turnover number, okay, so that's going to be the turnover number right there, is going to be the maximum number of substrate molecules that one molecule of enzyme can convert to product per second. The Michaelis constant Km is the inverse measure of the substrate affinity for the enzyme. A small km indicates high affinity, okay, so small km, high affinity of the substrate for the enzyme, and a large km, the opposite, obviously. At V equals 0.5 Vmax, half the enzyme active sites are bound to the substrate and the other half remain unoccupied. Therefore, the concentration of the enzyme equals the concentration of the enzyme substrate, and at some time, concentration of the substrate equals Km, so the dissociation of the enzyme substrate complex can be described as basically Kc, or the catalyst, equals the Km, where Km is the equilibrium constant of the ES, or the enzyme substrate complex. In other words, Km is equal to the dissociation constant of the enzyme substrate complex. Constants Km and Vmax also are quite useful to determine which of the competitive or non-competitive mechanisms are being used in terms of enzyme inhibition. So competitive inhibition can be overcome by increasing the number of substrate concentration and preventing the inhibitor from binding to the active site. As a result, the Vmax value in competitive inhibition remains the same, which you see here with the blue line. Okay. While the Km value increases as it takes more time for more substrate to reach the 0.5 Vmax reaction rate. So it just takes longer. So that's just, it gets there, it just takes longer. In contrast, the binding of non-competitive inhibitors to allosteric sites is not affected by the substrate, so Vmax will decrease. Oops, that's the green line there. Okay, a better line than that. Okay, so Vmax will decrease due to less effective binding of the substrate to the main active site, but at the same time, the 0.5 Vmax value will decrease proportionally to Vmax. So Km will not be affected by non-competitive inhibition, which again you see on the graph. So here's a problem. The kinetics of an enzyme catalyzed reaction are studied in the absence and presence of an inhibitor. The graph represents the initial rate of the function of a substrate concentration. So there's the graph. Identify the type of inhibition that's shown from the graph, so you could probably look at the previous slide. Determine Vmax and Km in the absence of an inhibitor and the presence of an inhibitor using graphical method, and outline the relationship between Km and enzyme activity. Okay, so it's going to look like it's going to be non-competitive inhibition from the shape of the graph. Absence of inhibitor, Vmax is 4.4, Km is 1.7, and in the presence of the inhibitor from the graph, Vmax is 3 and Km stays the same at 1.7. The higher the value of Km, the lower the activity of the enzyme. The lower the value of Km, the higher the activity of the enzyme. Okay, so you can have both arguments. The detection of proteins and the determination of their concentrations in solutions is known as a protein assay, and is the most common analytical procedure in biochemical experiments. In modern laboratories, protein assays often involve absorption spectroscopy in UV or visible regions of the EM spectrum. This technique is often referred to as UV spectroscopy, and it measures the absorption of UV and or visible light by proteins with organic dyes and transition metal ions. Almost all proteins absorb UV light around the value of 280 nanometers due to the presence of aromatic rings in particular the amino acids phenylalanine, tyrosine, and tryptophan. Certain organic dyes bind to arginine and aromatic residues and form highly conjugated systems of delocalized electrons, with a maximum absorption around 595 nanometers, which is kind of like orange really. The complex of proteins with transition metal ions also absorb visible light due to d orbital electron transitions. So you can see from the slide a typical UV-Vis spectrophotometer, and it consists of a light source. Here we go, the light source that produces the UV and visible light, a monochromometer that allows only, okay, that's kind of that there, a monochromometer which only allows narrow bandwidth of light to pass through, a cuvette, okay, so that's that kind of, that's what we call the container, a cuvette that holds the studied sample, a detector, okay, photocell on this example here, and an amplifier that convert the light into an electrical current, and then a digital output, so you can see some numbers. The sample solution containing a protein is put into a transparent cuvette and placed inside the spectrophotometer, and depending on the protein concentration and the experimental conditions, the intensity of the UV or visible light passed through the sample will reduce to some degree. And then we can use some fancy maths. Okay, and that fancy maths involves the Beer-Lambert law. Okay, there is a logarithmic ratio between the intensity of light emitted by the monochromometer, okay, I naught, that guy there, the initial, okay, and the intensity of light passed through the sample I, and this is known as the absorbance, capital A. Okay, and there's the equation there. Okay, it's going to be log base 10 of the I naught divided by I. The concentration C of the protein in the sample solution can be determined from the absorbance, therefore using Beer-Lambert's law there. Okay, where L is the cuvette length and the epsilon is the constant known as the molar absorptivity or the extinction coefficient. I tend to use the term extinction coefficient, and that depends on the solvent nature and the temperature of the associated solution. If the same cuvette and experimental conditions are used, the product of epsilon, or the product of the extinction coefficient and the length, will become a constant, so the protein concentration can be determined by the calibration curve of concentration versus absorbance, because that becomes a constant and that becomes a constant. So essentially A is equal to C. Okay, so to identify a particular protein, you first need to be separated out by something known as high performance liquid chromatography, and is then compared to a database with information from either a whole range of information from UV-Vis spec to gel electrophoresis, proton MR, and mass spectrophotometry, which we've already looked at. So just to finish up, here is a problem. The following tubes were set up. There are the results. So complete this column of concentration versus protein for tubes 1 to 6 in the table. Plot a wonderful calibration curve, and then use this curve to calculate the protein concentration of the unknown sample. That'll take a bit of work, so pause the video now, and then we can come back and see if you got the correct answers. Okay, so these are the answers. You should have got that there for the first one, plugging in your numbers there, getting concentration of the protein in milligrams per centimeters cubed. Have a nice, well, not quite straight line, because you know it's kind of curves up here. That's the problem with the Beer-Lambert law. You have saturation problems, but you can get a nice reasonable straight line out of that. Okay, and that gives you the answer that the concentration of the protein is around about 0.75 milligrams per centimeters cubed in the sample tested. And that, in a nutshell, is the HL component for enzymes and proteins.